The Selfish Gene by Richard Dawkins, Review and Scientific Critique. Richard Dawkins' The Selfish Gene is a book on evolutionary biology, where Dawkins introduces a gene-centered view of evolution. He argues that genes are the primary units of selection, functioning in ways that promote their own replication, and that this selfish behavior explains the apparent design and complexity in living organisms. While the selfish gene has significantly influenced understanding of evolutionary biology, its core argument, that random mutation and natural selection alone can account for all aspects of life's complexity, falls short in addressing the requirements of complex specified functional information, irreducibly complex systems, and orphan genes and proteins. Dawkins' materialist approach lacks sufficient empirical grounding in these critical areas, resulting in a reductionist view that oversimplifies the true complexity of life. 1. Central thesis and the gene-centered model of evolution. Dawkins's central thesis is that genes act as selfish entities, striving for their own survival by driving the behavior and structure of organisms. In Dawkins's view, natural selection operates at the level of genes, with organisms serving as survival machines built by genes to further their own propagation. By framing genes as agents in this process, Dawkins promotes a model where random mutation and selection can account for all aspects of life's design without requiring any guiding intelligence. However, this approach introduces several problems, particularly when addressing the emergence of higher levels of biological complexity. 2. The limits of microevolution and low-level macroevolution. Dawkins' model effectively describes microevolution, adaptive changes within species genus-family boundaries, and, to an extent, low-level macroevolution, or speciation within genera. Empirical evidence from studies of antibiotic resistance, selective breeding, and observable environmental adaptations supports the role of mutation and selection in driving change within these boundaries. However, Dawkins extends these processes beyond their evidentiary limits, suggesting that they can explain all of life's complexity, including the emergence of new phyla, classes, and orders, naturalistic megaevolution. Limits of micro and low-level macroevolutionary mechanisms. While microevolutionary adaptations and low-level speciation occur through natural selection acting on genetic variation, these processes do not generate the entirely new, complex specified functional information required for life's higher forms. Low-level macroevolution may include speciation within a genus, but typically involves variations within pre-existing genetic frameworks or loss-of-function mutations. In contrast, megaevolution, such as the origin of new body plans, complex organs, or higher taxa, requires large amounts of new, specific information that natural selection and random mutations alone have not been shown to produce. 3. Dawkins' model fails to address complex specified functional information. Dawkins' gene-centered model does not account for the origin of complex specified functional information, which is necessary for the intricate and coordinated processes observed in biological systems. Complex specified functional information refers to highly functional and complex, interdependent arrangements required to fulfill precise biological roles. For example, cellular machinery and metabolic pathways rely on precise sequences of DNA and protein interactions, which must be arranged in specific ways to perform their functions. Random mutation as an insufficient source of functional information. Mutations predominantly lead to neutral or deleterious changes, and beneficial mutations involve minor modifications within an existing framework of adaptation within a species or genus, rather than the creation of new complex structures needed for the higher taxa, such as kingdom, phylum, class, order. Generating functional proteins, regulatory sequences, and entire systems of interdependent structures through random mutation and selection alone presents substantial probabilistic empirical defeaters. Dawkins' model does not provide an empirically demonstrated or empirically possible, testable, and falsifiable step-by-step biochemical mechanism for the creation of the specified functional information that is required for entirely new systems or complex structures. Intelligent design as a viable source of functional information, specified complexity, is characteristic of designed systems, where components are organized with purpose and functional dependence. Intelligent design posits that the complex information seen in life's higher forms indicates an intelligent source. The specified information content in biological molecules, analogous to coded language, aligns closely with intelligent causation and not with unguided random processes. Dawkins' rejection of design dismisses this hypothesis without fully addressing the probabilistic and empirical defeaters associated with creating specified functional information through natural mechanisms alone. 4. Failure to address irreducible complexity. Dawkins critiques intelligent design, but does not provide any step-by-step testable and falsifiable gradualistic biochemical model or viable pathway to address the reality of irreducible complexity, a key argument for design in biological systems. Irreducible complexity refers to systems that require multiple interdependent parts to function, where removing any part results in system failure. For instance, structures like the bacterial flagellum or the blood clotting cascade require all core components to be present for functionality. Problems with stepwise selection for irreducibly complex systems. In an irreducibly complex system, random mutations would need to produce each necessary component in a specific sequence to achieve functionality, but selection cannot act on intermediate forms that lack functionality. Dawkins proposes that components of such systems could evolve independently and later combine into a functional unit, but this explanation is wishful thinking and is not supported by empirical evidence or any testable and falsifiable step-by-step biochemical explanation or model. The lack of experimental data demonstrating a viable stepwise pathway to irreducibly complex systems defeats Dawkins' claim that these structures could arise solely through mutation and selection. 5. Dawkins ignores the existence of orphan genes and proteins. Orphan genes and orphan proteins, unique genetic elements with no known homologs or evolutionary precursors in other species, present a significant defeater to Dawkins' gene-centered model. These genes often play essential, species-specific roles, and their sudden appearance does not fit within a gradualist model of evolution. The existence of orphan genes, which are not derivable from pre-existing sequences through mutation and selection processes, raises defeaters against the naturalistic origin of novel genetic information. Inadequate explanation for the origin of orphan genes and proteins. Orphan genes appear in specific lineages without any evolutionary ancestors, which contradicts and defeats Dawkins' gradualist model. Dawkins' reliance on cumulative selection does not address how entirely new, functionally essential genes arise without any evolutionary precursors. Intelligent design provides a better and more rational alternative, positing that anything more than 10 orphan genes in any taxon are the result of intentional design rather than random emergence, aligning with the view that unique, functionally specific genes point to a purposeful origin. 6. Philosophical bias against intelligent design. Dawkins' approach to biology is strongly materialistic, with an a priori commitment to exclude intelligent design as a possible explanation for biological complexity. This philosophical bias constrains his analysis, preventing him from fully engaging with empirical findings that indicate design. By beginning with the assumption that all complexity must arise from undirected processes, Dawkins precludes a valid hypothesis without addressing its scientific merits. Intelligent design as a hypothesis for biological complexity. Intelligent design offers a scientific and rational framework for interpreting observed complexity in biological systems. It does not oppose scientific investigation, but proposes that certain features of life, such as specified complexity, irreducible structures, and unique genetic elements, point to an intelligent cause. Rather than dismissing intelligent design out of hand, a comprehensive scientific approach would consider it alongside materialistic explanations, particularly when the empirical evidence for naturalistic mechanisms falls short, and yet is consistent with purpose and design. 7. No testable pathway for irreducible complexity or functional information. Dawkins' model lacks a step-by-step, testable, and falsifiable biochemical pathway for how functional irreducible complexity or specified functional information could arise solely through mutation and selection. Scientific investigation requires experimental or observational data to support claims. Yet the atheist claim of formation of irreducible systems and the origin of specified functional information through random mutation and natural selection remain nothing more than wishful thinking. There are no experimental demonstrations in the scientific literature that show the gradual formation of functionally complex nanomachines by random chance and natural selection, or the emergence of specified information-rich sequences without an intelligent source, absence of experimental evidence for naturalistic mega-evolution, the lack of a testable biochemical mechanism for generating complex specified functional information, irreducible systems, and orphan genes highlights the limitations and defeaters of Dawkins' model. Naturalistic mega-evolution, particularly the formation of new body plans or higher taxa, requires more than minor modifications within existing frameworks. It necessitates new complex functional information, which random mutations cannot generate. Without a stepwise, experimentally verified biochemical pathway, Dawkins' reliance on mutation and selection as explanations for all complexity is nothing more than wishful thinking. Conclusion: the selfish gene popularizes a gene-centered view of evolution and highlights the role of genes in shaping biological functions and behaviors. However, Dawkins overextends the power of random mutation and natural selection by suggesting that they can account for all biological complexity, including the emergence of new body plans and higher taxa. Empirical evidence supports microevolution and low-level macroevolution within defined taxonomic limits, inside a species, genus, and taxonomic family, but does not substantiate the claim that these processes can produce the complex specified functional information, irreducible systems, and orphan genes necessary for naturalistic mega-evolution. Dawkins' philosophical commitment to materialism leads him to dismiss intelligent design without adequately addressing the defeaters posed by specified information, irreducible complexity, and unique genetic elements. Intelligent design, by contrast, provides a viable scientific hypothesis for the origins of life's complexity, showing that purposeful causation better explains the observed organization and information content in biological systems. Moving beyond Dawkins' reductionist view allows for a fuller scientific inquiry into the origins of complexity, one that considers all available evidence, including the reality of an intelligent cause. Thanks for watching. Please like, share, and subscribe. And comment below if you agree or would like to add your thoughts. If you would like me to respond to your comment, please make a Level 1 comment directly below the video. I will respond to all polite Level 1 comments even if it takes me a while due to other commitments.